It is my distinct pleasure to introduce Professor Nick Martin, who is joining us from Brisbane, Australia. Nick is, I think, the most consistent workshop faculty attendee, having yet to miss a workshop. And so we're really delighted to hear his perspective on the science that he was excited about when he was starting out in trying to understand the nature of the relationship between genetic variation and human phenotypic variation. So Nick, take it away.
Thank you, Ben. So what I wanted to do this morning was just go through 3 papers of historic interest now, which I think gives some important background to persisting themes in the field. It's somewhat egotistical because they all happen to be by me. But that aside, I hope you'll be interested. So there are 3 questions I sort of came into the field with when I was an undergraduate way back in the 70s. Firstly, how big do twin studies need to be? Secondly, what happens if twins are not sampled at random from the population? And thirdly, and perhaps most importantly, how do we unravel and estimate the causes of covariation between traits? So the first of these, how big do twin studies need to be, we address in this paper in 1978, and I have to pay huge homage to my PhD supervisor at the time, Lyndon Eves, and my 2 other colleagues here. I was really, in many ways, just the amanuensis, the annoying gnat, who nagged them until we got the answer out. So let's just begin with the well-known ACE model for twin data, in which we have 3 sources of variation we'd like to estimate the importance of: additive genetic variation, common or shared environment C, and unique environment, which we must never forget includes measurement error. So we'd like to estimate all 3 of these. And I mean, here's an example of applying this to some data we collected here in Australia on exam results, where we get results fairly typical of others, about 80% of variation genetic, about 10% due to error or non-shared environment, and 12% due to shared environment. But the question is, until the 1970s, most twin studies were very small, sometimes only dozens of pairs of twins, and people were publishing papers with results that were just all over the place. Some people would find large estimates of heritability, others no heritability, suggesting everything was due to shared environment. So the answer, why to this? Well, the obvious solution was that these had low power, but people weren't really thinking in those terms much in those days. And that led me nagging Lyndon to say, well, how big do twin studies need to be to give us consistent results and estimates of these variance components that we're interested in? And the answer turns out to depend upon the true sources of variation. Well, that's not particularly surprising, and the ratio of MZ to DZ twins in your sample. And so the way we went about it was to ask the question, given that we know the true components of variation contributing to the population variance, we want to know how many pairs of twins are needed to be 95% sure of rejecting alternative wrong models at, say, the 5% level of significance. In fact, you can choose which numbers you want here, and I think people nowadays would work with 90% or even 80% sure, which is the power, and the 5% level of significance, I guess is still pertains for most people, depending, of course, on the number of tests that you do. So our procedure was to take the right model and obtain the expected covariances, in quotes, for given values of population parameters, then obtain maximum likelihood solutions for the parameters of the wrong model that we fitted to those data, to obtain the weighted residual sum of squares, or the chi-square, for the fit of the wrong model, and use this as what is called a non-centrality parameter, lambda primed, for S minus P degrees of freedom, where S is the number of statistics, If you've got MZ covariance, DZ covariance, then you've only got 2. And there are P parameters in the wrong model. And those parameters can be A, C, E, any combination of those. And then we can look up that non-centrality parameter in tables. We used to have to look that up in paper tables by Pearson and Hartley, but nowadays you can easily do it online. And then obtain the required sample size, dividing that lambda by lambda prime. So these were the tables we produced in that paper back in 1978, and they take a little bit of absorption. First of all, the notation's changed since then. In those days, we referred to E1 and E2 for what we now call E, unique environment, and C, shared environment. And we referred to VA, VA, what we now call A, we used to call VA. And then, so down the y-axis here, we have, in our true model, different proportions of unique environment and shared environment. And then for the sample, the models that we're going to fit, firstly, just a straight E model, and secondly, an EA model. And these are the 2 models we've got most frequently fit. We can then look at the numbers of pairs we would need in our sample, given these different proportions of monozygotic twins in our sample, going from 10% up to 90% MZ. In fact, the population proportions around here. So, and most twin samples generally have, somewhere in this area. So that's the sort of area you need to be looking at. And anyway, you can see that in this first case of fitting an EC model or CE model to these data, you don't actually need that many pairs to reject a false model that says, well, actually all variation is just due to unique environment, or indeed a model that has both E and A. Although the numbers are starting to get up there, even for that case. Then if you look at the reverse of that situation, where your true model in the population is the true sources of variation really are E and A in these varying proportions. And then we try and fit an incorrect E model or an incorrect CE model. Then you can start to see that the numbers we would need. So let's take a case here where the true population, proportions of the population are about 50% E, 50% A, which happens to be about the case for personality, for example, and many other psychiatric symptom traits. Then you can see that the numbers we require to estimate just an E model starting to get up there, but to reject a CE model, you need quite large numbers. You're already getting up to, 800, 600 pairs of twins. So, I mean, that's telling you that most of the studies done up to that time, 1978, were nowhere near that number. And even for this modest case, there was really no hope of getting clear results. Now, if we then get more complicated and go to this, I'm afraid, horribly dense slide, where we have all 3 sources of variation. Incidentally, I'm not talking about dominance here. That's a quite different case again. But if we just take this case where we've got both E and C and A, and then try fitting false models of E, CE, and AE models, as we'd now call them, you can see the numbers start to get a bit more frightening. So let's take one case here. So we've got a trait that's 30% E, 30% C, and 40% A, which just happens to be about the story for social attitudes, which is a trait that's been of interest to me, but also many other behavioral traits. Then to fit an E model, you're very well powered, no problem. But to reject an CE model, you can see you need pretty large numbers. And likewise, to reject an incorrect AE model that omits C, you also need pretty large numbers. So this was really a surprise when we did it. I don't think we had any idea of the sample sizes that we would need to be able to get discrimination. So what's the point of all this? Well, the point let's just summarize what I've said. For equivalent cases, it's always found easier to reject an inappropriate simple genetic model, an AE model, than an inappropriate simple environmental model, a C model. And the optimum proportion of MZ and DZ twins in a sample will vary with the true model, but is most likely to be around the natural European proportion. So that's sort of quite good news for those of us actually doing these studies. That for several frequently encounters, we need large numbers, more than 600 pairs of twins in our study. And by the way, if you want to do this separately for males and females, which I always think is advisable, because you can never assume that the sources of variation are the same between males and females, then you're going to need 600 male pairs and 600 female pairs. And I haven't dealt with the case of dominance. Much of our paper, the one I showed you, actually deals with the problem of detecting dominance, but that turns out to be a much harder problem again, and requires more than 3,000 pairs. So the consequence of conclusions and consequences of this work were that twin studies really had to be very much larger than they had been up to that time. And this led directly, in my own case, to the foundation of the Australian Twin Register, in which we recruited 15,000 pairs of twins in the space of about a year. And I know was a pretty strong motivation as well in the foundation of the Netherlands Twin Register by Direct Boomsma, and I think a stimulus to the formation and expansion of other large national twin registers, and indeed, trying to focus on how we could get large-scale systematic recruitment to these registries through birth registries, health systems, school systems, which is what we used in Australia, or whole population registries, which Scandinavia was particularly well equipped for. And this in turn led, even as into the late 80s and sort of going into the 90s, producing much more consistent estimates of heritability for the same trait between countries, between studies. And I think this really led to people outside the field starting to take it much more seriously. Because before that, people had been, oh, look, it's all over the place. How can we trust anything? Whereas now people had to say, well, actually, you do this study in the US, you do it in Australia, you do it in Scandinavia, and you're getting pretty much the same result. Maybe there's something real here. And I don't think it's too much of a stretch to say this really set the scene for efforts to find the actual genes, which started off with candidate gene studies, which a lot of people wasted time in the 80s on, and I think generally regarded as a complete failure, almost complete failure. Linkage, which many of us, including me, invested hugely in the 90s, which also one has to say in retrospect was a total failure. And it was the failure of those that actually led to the development of genome-wide SNP chips, which has produced the outstanding success we've seen since 2005. So I do like to think there's a sort of a linear progression in all of this, which has been useful. Another sort of side effect, which some people curse, is that power calculations as above are now standard in all grant applications if you want to get have any hope of getting funded. And rather than wade through our laborious things, which Mike Neal, amongst others, have found some small technical faults with, I don't think in principle there's anything wrong with them, but there's just some wrinkles there that are interesting. But anyway, it's all been made much easier by this wonderful set of tools that Sean Purcell has made available on his website, and which I'd recommend to you, particularly if you're writing a grant. Okay, second paper. What happens if twins are not sampled at random from the population? And this problem came up from my own work. I did it as an honours thesis, which was actually looking at trying to do genetic analysis of exam results in the South Australian population where I grew up. And there was an exam that was taken by about 2/3 of the age cohort, and one can safely assume it was the more able 2/3 of the sample. And so I spent my honours year actually collecting all those twins and finding out their zygosity and calculating the heritability of all of these examination performance traits. And then having got that, it occurred to me, well, the trouble is that that is actually only from this truncated sample of the population, only from the top 2/3. And what would the results we've got imply for the whole population? Because our subsample is certainly not representative. So what we did was, first of all, to ask, well, if you have truncation selection, and these are just normal deviates where the selection threshold is applied. Sorry, I should have had a pretty figure for this. We had just asked the question, well, if you've got one twin who's in a sample, what's the probability of the other twin being in the sample if you've got truncation selection? So if you have selection if you leave out the bottom 10% of your sample, then you can see that for various situations, you've only got about an 80% chance of having your co-twin in the sample. If you get over to here and you leave out the top, you've got only the top 10% of your population sample. The chance of having both twins in your sample is very small. It's well less than 10%. And everything else is in between. So that tells you that this is going to have some likely major effect. So if you now calculate the correlations for those samples, given these different, where these are the true population values of the correlation without any selection at all, but that you then impose these varying degrees of truncation selection. So you're only taking people above these limits, so below these limits, then what is the consequence for your correlations? We can see with only 10% truncation, it's not too bad. 30%, you can start to see this sort of differential effect between the cases for these different true correlations. And that gets worse as you get over there. And in fact, this is very much similar. This is very much the case I had in my own study. And so, in fact, from my own study, we were just on the raw data estimating MZ and DZ correlations of 0.8 and 0.4, which were consistent with a heritability about 0.8 and no shared environment, which was very surprising given that we know that there's strong assortative mating for cognitive variables. And in twin studies, assortative mating looks like C. That's another whole lecture on that topic. I'm sorry to just skim over it like that, but it's an important point, which I hope someone else will cover. But interpolation amongst the curves in figure 1, which I've already just shown you, would suggest that the population estimates are actually, the real estimates are about 0.9 and 0.6, which are consistent with a random mating heritability of about 0.6 and a between families component of 0.3, which is compatible with a considerable degree of assortative mating. And in fact, we can make a post-hoc adjustment for that, which one can do. Anyway, so this has become very important. I mean, this I think was the first stab at this problem, and has led Mike Neal in particular to really develop some very, very clever ways of tackling this problem in MX. And in particular, the implementation of what is called full information of maximum likelihood, FIML, which is the standard application in MX and makes use of all the data collected. I should say there's a huge body of theory behind this called missing data theory, or missing at random, missing completely at random. Mike gives a very eloquent lecture on this. I hope you'll get to hear it. But anyway, that from a practical point of view, FIML makes use of all the data collected, including both complete and incomplete twin pairs, and you must include those in your sample. And I can't give you the full mathematical theory, but at a sort of verbal level, what FIML is doing is making use of the difference in mean and variance between complete and incomplete pairs and comparing those for both MZ and DZ twins. And you'll see for traits that are genetic, you'll actually see differential effects there between the MZ and DZ pairs. And Phil makes use of those to estimate the true population values of means and variance components. So if you use FIML, this problem just gets solved for you, which is neat, but you need to be aware of it. And always make sure you put the same effort into collecting incomplete pairs as your complete pairs. And I mean the same effort. You don't put more effort or less effort. You put the same effort, because otherwise the sampling theory doesn't hold. Okay, enough on that. All right, the final paper, which in many ways I think is the most interesting to behave geneticists, is how do we unravel and estimate the covariation, the cause of covariation between traits. And we all, in our studies, deal with traits that are correlated, and we look at the correlation and say, I wonder why these are correlated. This is because all of these are caused by the same genes or the same environmental factors or some combination of both. Or could it be that trait A causes trait B or vice versa? So there's a whole, this is the whole, I guess it's almost the core of biostatistics in our game. We try to understand the causes of covariation between different traits. And obviously this problem had been out there for a long time. By the time we came along to it in the late 70s, and there'd been some very notable efforts to solve it, notably by Stephen Vandenberg, who used to be a founding member of the Institute for Behaviour Genetics in Boulder, by Lyndon Eves and another lecturer called Jeff Gale in Birmingham, and John Lowland. All these people had a go at it, and most people were sort of trying to tackle it in two parts. One was to estimate the genetic, the different covariance matrices for genes and environment, and then to apply factor analysis to it, or to do factor analysis of your data and then do variance components analysis of those factor scores. And what we want, and we could see the limitations in both of those, because whichever way you did it, one would affect the other. With biases that were not clear. And so what we wanted was a solution where you did it all simultaneously to obtain the maximum likelihood solution for both the sources of covariation and their structure, their factor structure. So this slide's a bit of a horror, and I do apologize. You have to be able to think in matrices. But to those of you who've done psych, you should at least be familiar with this factor model. Sigma throughout here is our expected covariance matrix, which for the factor model is a function of a matrix of factor loadings for V variables and F factors. So it's a V by F matrix. And phi is a matrix of correlations between those factors. So if we want to specify that our factors are orthogonal or independent of each other, then phi will just be an identity matrix, but it needn't be. You can have a sort of Promax rotation or something where the off-diagonal terms are non-zero. And out here is our old familiar specifics. So these are specific causes of variation that are not summarized by the factor structure. So that's where we begin. And everything that we did was derived from the brilliant work of a Swedish statistician called Carl Joreskov, whom we actually persuaded to come to several of the early workshops because we thought he was just such a great guy. And he extended this this model to actually, we elaborated it to put outside that factor model, this beta matrix, which enabled you to scale all those variables in a variety of ways. And there's something that we in the field have made use of to enable us to fit things like the common pathway model, but most particularly develop the theory of directional causation modeling, which you'll hear about in the course. This theta squared is another set of specifics, but we needn't talk about that. Anyway, our contribution to the field, and I have to say this is entirely the insight of Lyndon Eaves, as I say, I was just his amanuensis really, was he just left stuff all over the floor and I went around picking it up and sort of trying to put it in order. Anyway, so here's Yoroscogg's covariance structure model. What Lyndon realized is that we could simply take that and then for each covariance matrix, and that's so summing across covariance matrix for MZ twins and for DZ twins, and then we could specify, well, first of all, for each of our sources of variation, A, C, and E, which are P sources, so for each of those J sources, P sources, we could specify a covariance structure matrix, and then we would sum those with their appropriate coefficients from the ACE model across our MZ and DZ matrices to get the expected covariance matrix. So that was the real insight, which we then programmed in Fortran rather lugubriously, but it seemed to work. But people people were interested in it, but that didn't take off because nobody could work our programs and because the notation at that time was pretty clumsy. So just to finish that off, so this development enabled us to really tackle multivariate genetics. It's enabled us to tackle time problems and to look at other problems in behavior genetics, like the sources of assorted mating, interaction between spouses and other family members, and the transmission of culture down through pedigrees. But as I say, the problem was that the notation was awkward, and it was other people in the field, notably David Fulker, but others, George Vogler, I seem to remember, who made the breakthrough in the early 80s, translating our rather clumsy notation into the terms of path analysis, which had been developed for animal breeding by Sewell Wright back in the 1920s. And in fact, colleagues in St. Louis, Cloninger, Rice, and Reich had also already seen, and Newton Morton had already seen the value of. And it was by translating our ideas into terms of path analysis that we were able to tackle, tackle a whole series of multivariate problems. This is just a simple factor model, elaborating the earliest univariate slide on looking at exam results and the influence of verbal IQ and performance IQ on those. And this is another schematic example of how one applies the same sort of theory to developmental change, so to longitudinal analysis. And that's just a brief sample of two, but the course is going to be about this largely, so I won't waste more time. I just simply wanted in this brief talk to give you some idea of where some of these ideas came from. Thank you very much.
Thank you, Nick. That was amazing. A few questions.
How long was I?
Here.
Have a breather.
Well, yeah. Do you want a breather, or do you want to stop sharing and then we can chat face to face? Maybe if that's a good idea.
Yeah. I'll stop share.
Okay.
Yeah.
So just a few questions. I was struck by a few things. One is it almost felt like the way you were describing the power of the classical twin design, like the field was gripped in a reproducibility crisis. Is that, is that the sort of sense of the time?
To put it in 2021 terms, that's exactly what it was, yeah. Everyone's talking about the reproducibility crisis. When I hear it, I just yawn and think, my God, I've been there before. And our subject, behavior genetics, had that very, very much in the 60s and early 70s. And for those of us who cared about the field, this was distressing because we knew there was something real there. We just needed to do something about it. So that's what drove that whole endeavor. I have to say, another major stimulus that I didn't mention was the NMSQT twin study by R.C. Nichols and John Lowland, which they published. The NMSQT is a National Merit Scholarship Qualifying Test. And Bob Nichols, who was involved with that, managed to get hold of the records and extract all candidates for that exam, which incidentally was probably taken only by about the top 10% of the population, and get all the candidates for the same surnames and birth dates and pull out the twins. And that sample was, from memory, close to 1,000 pairs. Last one started to get results that seem meaningful. But once again, I don't think any of those estimates of heritability came with standard errors or any sense of what you did and did not have the power. But I think that was it was an inkling of the fact that we needed to get bigger, and that was something else that drove us on.
But I also liked the sort of rigor that is baked into this, and also the pieces of work that you point to. I mean, as I said, across all three of them, they are as much about how we do science as they are about any discovery relevant to a specific data set. And I think that that's like a meaningful contribution as well. It's a meaningful way to look at the way science is not just simply a data set that you're analyzing. It is a philosophy, an approach to how you think about interpreting data and how you think about collecting and looking at that data.
Well, Ben, I'm sure you're the same as I. You got a bright student who bounces up to you with a great idea or a great piece of work or something, and say, "Fantastic, great. Now I go away and try and destroy it."
Yep, exactly.
Because that's what science is about. Science is, as Popper taught us, is about falsifiability. And it's always trying to find the weakness in what are the weakest links in the chain, and what can we do about them? Mm-hmm. And so these pieces of work was certainly the first two were very much in that. In that philosophy. Well, actually, so was the third one, because it was a dissatisfaction with the sort of rather ad hoc methods that were being used to try and take apart the causes of covariation in these two-step ways. And it was absolutely obvious that the order in which you did those two steps and how you did them, that would affect the second step, and that just wasn't satisfactory. Of course, it doesn't mean that the maximum likelihood solution that we get is the best, is right. But I mean, it's probably, I think it's probably the best that one can do.
Well, it's the maximum likelihood, and that's the best estimator that you can get to if your likelihood function is well conditioned. Yeah. So
so
sure.
I think,
sounds
good.
It
was a quest for maximum likelihood.
Mm-hmm. Mm-hmm. Do you know what a Heckman correction is?
No, I don't.
Okay, so Heckman correction was ascertainment bias correction that Heckman, an economist, figured out and won the Nobel Prize in economics in 1974 for. So what you were doing with the second paper was very similar to what was being shown in the Heckman correction paper about biases and estimates of statistics as a function of ascertainment biases in your cohort. So that's Nobel Prize winning work there, Nick.
Well, of course, the other person who had a go at this before us in the plant breeding, I mean, this really comes out of Kemp Thorne's work on truncation selection. It's very similar to that. But I think what complicated the case we tackled is bivariate, and that there are two twins. Yeah. And you're not just losing an individual, you're losing a pair. And of course, that was the brilliance of Mike's solution of how we tackle this with Fimmel, that if you actually assiduously include all of the incomplete pairs, and you put the same effort into following those up as you do your complete pairs, then you can actually correct for this. And the simulations all back that up beautifully. So that was a a really beautiful piece of work. I don't think ever really got, I mean, to me, that just sort of crept in the back door of the workshop. I don't know if there's ever been a paper that sort of heralded that, but to me, that was a wonderful piece of work.
And on the topic of the genetic covariance, when you cracked it, did you know? When you saw, when the insight was there, did you know?
Yeah, we did. I mean, and I was so excited. I was so excited. And I remember, and this is maybe a lesson for some young people listening. So we went along to the Genetical Society. It was called the Genetical Society of Great Britain in Cambridge in 1977, which was at St. John's College. Beautiful setting. I'm sure many of you know it. And I presented this to the Genetical Society. And it was just, as Lyndon Eves would say, a stone down a well. There was not one question. There was not the slightest reaction. No one had a clue what I was talking about. No one got it, you know, pass on by. I think I went out and got roaring drunk that night. But yeah, so that was a great disappointment. But my father tells the story, who was a geneticist as well. He tells the story of being one of the first in his department, you know, at our end of the world in Adelaide, to read the Watson and Crick paper in 1952 or 3, was it? And present that as a journal club, as we now call it, to his department. Same thing, complete stone down a well. No one got it. No one could see any points or whatever. So I think this is not uncommon in science.
Yeah. It's hard to capture people's imagination, but you know when you're working on a problem that you've actually made progress. Like that moment, it's real. It's there. Well, I mean, you can feel it.
Lots of other people have talked about this. Mathematicians talk about it, and I, you know, I'm no mathematician, but there's a sense in which you know you've got something that's very elegant. Yeah. And I mean, all you can do is look at that equation.
Yeah.
And it's all there. It's all there. Mm-hmm. And, you know, and it's that, you know, that's elegant. And it's got to be right.
Yeah, and I mean, as simple as possible and no simpler, right?
Yeah.
Okay, Nick, any closing thoughts or things you want to tell to the students? It's been really wonderful hearing you tell this history. I mean, it really is foundational work for the community. And I think the uptake of the ACE model speaks to the impact of Martin and Eves, shall we say, in the scheme of things. So yeah.
Well, look, I guess I just would like to pay obvious tribute to the workshop and in particular, you know, the real mainstays of that. And Lyndon, I've mentioned quite a few times. It's unfortunate that young people don't have much inkling of how important David Fulker was, who was, you know, one of the real intellectual giants of the field and who died tragically of pancreatic cancer in 1999, I think it was. But, you know, he was an absolute mainstay and many of the developments in the subject and you know, how the concepts I've talked about this morning got morphed into much more elegant, efficient ways. And then, I mean, the tremendous work of Mike Neal and Airmean and making that accessible through MX and Airmean's beautiful PowerPoint displays and diagrams and so on, all of which just has made this much more accessible. And you just see the literature's just dominated by this stuff. So I think we've made a difference.
Well, it's been a pleasure hearing your story and hearing the oral tradition of the workshop and what's sprung from it and the academic tradition from whence it came. So sad that we won't be in person, but it's been lovely to see you and catch up later.